Okay, hello, my name is Shahab, and in this video I will be explaining to you the urea cycle. And mind you, I'm not going to be explaining the whole lecture or whatever is in the lecture, like deficiencies or anything that's in the beginning of the lecture. For those, you have to refer back to your slides, because in this video I'll only be explaining the cycle itself. I'll try to simplify the cycle itself, and so yeah. If Dr. Wissam is still teaching you, you might find something like this in one of your slides. This is the urea cycle. Now it looks horrific as of now, but with Dr. Wassim's explanation is really great. But I thought, instead of for you guys to have to look at this as a whole, why not break it down and go through the steps one by one, make it easier, make it simpler, because that's how I enjoy going through cycles. So we're gonna start with the very basic information that urea cycle mostly takes place in the liver, because the liver, and I say mostly in the liver because the liver is the only the liver cells are the only cells in the body which have all the enzymes required for a full urea cycle. That's why it mostly takes place in the liver. Now, what is the main purpose of the urea cycle? It's basically to turn ammonia into urea, so urea can be excreted by the kidneys out of the body as a waste product, because ammonia is also a waste product that is formed as a result of other chemical reactions within the body. Now, let's start with a simple idea. Where does the urea cycle take place in the cell? It takes place in two places, which are the mitochondrial matrix and the cytosol. The mitochondrial matrix is obviously depicted as the orange semicircle, and the cytosol is the green space. Now let's start with the first component in the 5-step urea cycle. This 5-step urea cycle starts with the component ammonia, okay, which is a waste product, as I said, of other chemical reactions in the body. Now, ammonia, in order to enter the urea cycle, it needs a carbon. Where does the carbon come from, you ask? It comes from CO2. And in order for ammonia and carbon to mix and enter the urea cycle, they need help from the energy currency of the cell, which is ATP. So in this phase, it uses 2 ATP to turn into carbamyl phosphate. Now, now this reaction, every reaction in the body needs an enzyme, or almost every reaction in the body needs an enzyme. For this reaction, what enzyme helps the reaction take place? Carbamyl phosphate synthase 1. Why 1? Because there is carbamyl phosphate synthase 2. Now, carbamyl phosphate synthase 1, from its name, it basically synthesizes or helps in the production of carbamyl phosphate. That's how one easy way to memorize the name, the enzyme, which the enzymes of this cycle are very important. Now, obviously, every reaction also has byproducts, or almost every reaction has byproducts. The byproducts in this reaction include 3 hydrogen atoms, 2 ADP, which are the result of ATP, and finally a inorganic phosphate. Now, in the mitochondrial matrix, the carbonyl phosphate stumbles upon a amino acid. What is this amino acid? This amino acid is ornithine. Now, where does this ornithine come from? We'll get back to that later. But for now, all you have to know is that ornithine is found in the mitochondrial matrix. Now, obviously, carbonyl phosphate and ornithine react with each other, so they can form a product. So what do they form? Citrulline. Now, again, as I said, almost every reaction needs a enzyme. Now, what is the enzyme for this reaction? The enzyme for this reaction is ornithine transcarbamylase. So yeah. There is one thing I want to mention is when you see synthase, it means it's building. When you see lase, it means, or ase, sorry, it means it's breaking down. Now, citrulline has special transporters that transport it out of the mitochondrial matrix and into the cytosol. So remember how we said there are two reactions that take place in the mitochondrial matrix, and 3 reactions of the 5 reactions in the urea cycle take place in the cytosol. So the 2 reactions are the one that takes place the one reaction is overlooked by the enzyme carbonyl phosphate synthase, and the other reaction is overlooked by the enzyme ornithine transcarbamylase. And once these 2 reactions take place and the final product in the mitochondrial matrix is L-citrulline, a special transporter shuttles the citrulline out of the mitochondrial matrix and into the cytosol. Now, in the cytosol, we find the amino acid aspartate. Now, where did this aspartate come from? We'll also get back to that later. For now, all you have to know is citrulline and aspartate react with each other, and the enzyme that helps this reaction is called arginosuccinate synthase. Again, here synthase means building. And obviously, this is another step in the urea cycle that requires ATP and produces AMP and 2 inorganic phosphates as a byproduct. So what does aspartate and citrulline form? What do they form? Basically, they form arginosuccinate. Remember, here it's written here, as you can see, it's arginosuccinate synthase. So basically, what is it synthesizing? Arginosuccinate from citrulline and aspartate. That's another way. That's another easy enzyme you can link to the product, which can help you memorize easier. Now, arginine succinate is turned into arginine. Now, remember how I said whenever it's ase or lyase, it means it's breaking down. So argininosuccinate breaks down arginosuccinate to form arginine. And what is the byproduct? Obviously, when there is a breakage, there is always a byproduct. So the byproduct here is fumarate. Now, remember how we said aspartate is found in cytosol? This is how we find. This is I'll get to that through fumarate. Now, fumarate, with the help of some Krebs cycle enzymes that are found in the cytosol, turns into malate. Malate then turns into oxaloacetate. Now, oxaloacetate turns into aspartate with the help of glutamate, which deaminates helps in the deamination of oxaloacetate into aspartate, turning glutamate into alpha ketoglutarate, because this is a deamination process that is mentioned earlier in the lecture. If you go through your lecture slide, you'll find it in the earlier slides. You'll understand how this process happens. But in this cycle, all you need to know is that oxaloacetate, with the help of glutamate, turns into aspartate through the process of deamination. And glutamate turns into alpha ketoglutarate because of this process. So that's how we get aspartate in the cytosol, through lysing arginosuccinate into arginine. You bring out fumarate, turn it into malate, oxaloacetate, and then we get aspartate with the help of glutamate. Now, finally, arginine turns into ornithine. Now, what is the enzyme that is used in this process? Arginase. Ase means breaking down. So arginine turns into ornithine through the enzyme arginase. Obviously, with the use of water, you also get urea as a byproduct, which is, remember we mentioned that we want to turn ammonia into urea because that's the main objective of the urea cycle. So this urea, so the cycle keeps on going and going and going until the body has gotten rid of enough of ammonia and formed enough urea for the kidneys to get rid of the toxic waste that is urea. And finally, ornithine gets transported back into the mitochondrial matrix. Remember how we said in the beginning of this recording, remember how we mentioned that ornithine is found in the mitochondrial matrix? Well, this is how it is found in the mitochondrial matrix. It is the end product of the urea cycle, which is also helps in forming the beginning product, which is L-citrulline, that gets shuttled out of the mitochondrial matrix and forms the cycle. So this all comes around from the ammonia. From the ammonia, it goes into carbonyl phosphate. Carbonyl phosphate turns into citrulline. Citrulline, with the help of aspartate, turns into arginosuccinate. Arginosuccinate goes into arginine, and arginine goes into ornithine. Ornithine goes back into the mitochondrial matrix. And this is how the cycle is formed. This is why it's called the cycle, because it goes in circles. And yeah, basically this is the very brief, not really detailed explanation of the urea cycle. I hope this simplified the topic for you, and have a good day.